Yeah, you can start. Yep.
Yeah.
Yep.
Okay. All right. So, welcome back to this four lecture series on quantum biology, and today it'll be the fourth lecture, and I'll be talking about aspects related to the human brain specifically. And I want to start with a little commercial. I don't know if I told you about this before, but about 2 years ago, I co-authored a book on quantum body with two doctors, Deepak Chopra, who is a medical doctor by training and also public intellectual, I guess, and a public figure, and Brian Fertig, who is an endocrinologist from New Jersey. And I'm a physicist here in between two doctors. And today I will not be talking about this whole quantum body concept, which is expanded in the book from three different directions: the direction of medicine, the direction of physics, and the direction of spirituality, due to Deepak's major contribution here. Instead, I will focus just on the human brain, which is challenging enough and fascinating enough. And we'll delve a little bit deeper into this. Will be maybe less physics and more neuroscience. We'll be asking some questions, some not too difficult and some very difficult, and some which are unexplainable or unanswerable at the moment, at least. So these are the questions, I would say, not too tremendously difficult, but important questions. What is the computational power of the brain? We do, at least some of us do, computations in our brain. And we'll see that the brain consumption, although on the scale of the human body is significant, 25%. Energy, yeah. So, but it's tiny compared to technological devices performing computation, like computers and so on. And then this is a more profound question. Is information processing taking place in the brain classical, quantum, or something else? And the bet is on quantum, at least to some degree, at least in some aspects, yes. And then this is maybe less profound, but important, and I would say quite open to some degree. How are all sensory inputs integrated and synchronized as we observe reality surrounding us, and we see all of it and hear and sense and smell the environment all at the same time. There's no delay, there's no disconnect. So how does it happen? And I would say right from the start that mechanisms of computation based on or information processing more generally based on biochemistry would be too slow. So there has to be some physics, whether it's quantum or classical. It's to a large degree still to be determined. Let's start with the numbers. So we have 100 billion neurons approximately in the brain, and the same number of glial cells supporting neurons energetically, and some other types of cells, but we'll just focus on neurons today. And they are connected by synapses, on average about 10,000 synapses per two neurons. So it's on the order of 10 to the 15th, which is 1,000 trillion synapses. Even if we conservatively assume that they operate at about 10 impulses per second or every 100 milliseconds, I would say that's really conservative, maybe quite a bit faster, but even that gives us about the maximum operating speed of 10 to the 16th operations per second, which outperforms supercomputers such as IBM's Blue Gene with 10 to the 15th. That's already quite impressive, but what's more impressive is that with 25% of the energy supply for the entire body going into the brain, that translates into 25 watts. Compare it to the Blue Jeans supercomputer's requirement of 1.5 megawatts, so 1.5 million watts. So we are talking about 100,000 times or so less energy hungry, so to speak, or consuming, with a 10 times, possibly, potential 10 times larger number of operations per second. So this is quite amazing. And that number, if you want to put it in context, 25 watts, this is more or less, maybe even less than an incandescent bulb in your night desk lamp, which is typically about 40 watts for a lamp. And they can go to 60 watts in your dining room, or maybe 100. So this is a fraction of the power consumption of incandescent lamps. When you think about this, I think it's quite amazing that the brain can do so much so fast with so little energy consumption. But there are also big questions about the brain. These were big enough, but not huge, because this is what at least most of us, I think, believe, where consciousness resides. But what is consciousness, and where exactly is it localized in the brain, if at all? Next, how did it evolve during the evolution of life on the planet? Is it associated with all living systems? Is life equivalent to consciousness, or is it hierarchical? Maybe it's it has a higher level as you proceed on the evolutionary tree. Probably yes. Maybe there's some proto-consciousness associated with very simple single-cell organisms. Maybe bacteria have some consciousness, rudimentary. Then where is memory start? Where exactly is memory? And there are some challenging claims about this. Maybe not just in the brain, but in some other. Maybe the entire body has some form of memory. We talk about muscle memory. When you train something, you get better, and there is memory encoded in the functioning of the human body. I mentioned this already. There's also in between classical quantum, maybe fractal, which means that there would be some kind of hierarchy of information processing, stretching from quantum to classical through some scaling across the different length scales and time scales. I don't think I'll have too much time to elaborate on this, but that's a question that many scientists wonder about. And I mentioned this already. But these are the big ones. These three, four actually. This one too. But what are the brain's activities? Well, among other things, just communication across the human body and with the environment. So it has sensory inputs, receives inputs, and generates outputs, and in between it processes this information and decides on how to react to stimuli. Is it digital? Is it analog? Is this information electromagnetic? And we know it's action potentials. So there's some electric activity, to be sure, and that brings us to this very quick run through the neurophysiology era of time, starting with Leonardo da Vinci's modeling of various parts of the human body, and then Humphrey Ridley's anatomy of the brain, going back more than 330 years. And then we have the first connection to electricity. This is all anatomical. Now we're going into the functional. Luigi Galvani in 1791 published the work on electrical stimulation of frog nerves. And then the next milestone is, now it's almost 200 years ago, William Morton at Mass General demonstrated the use of ether for anesthesia, which allowed, of course, we talked about this at the end of the last lecture briefly, and we'll come back later on, a monumental discovery allowing all kinds of surgical procedures and other ones which would be possible. And then we go to the end of 19th century, where Ramon y Cajal in Spain provided very detailed illustrations of the various parts of the nerve system, including detailed graphical representations of Purkinje cells and all kinds of brain cells. And then another more than half a century later, we have the monumental discovery of these three scientists, John Eccles, Alan Hodgkin, and Andrew Huxley, who got a Nobel Prize for their mathematical explanation of the neuronal activation and transmission of electrical signals in what is known as the action potential. We call these Hodgkin-Huxley equations, or for HH for equations for sure, for short. And these are coupled nonlinear differential equations, which very accurately describe the shape and the speed of the electrical signal, the voltage changes across the neuronal membrane, propagating with speeds somewhere in the range from 0.5 to about 20 meters per second, which gives us sort of the reaction time on the order of a fraction of a second, which later on in the work of Libet corresponds to the delay time in our reactions on the order of half a second, 500 milliseconds, maybe less, something of this order. And the Hodgkin-Huxley model is really an electrical circuit representation of the neuron, and you can go deeper into it. And more recently, in fact, a few years ago, there was a demonstration that's not only just a voltage pulse that's propagating along the axon with these speeds, but also there's a mechanical deformation of the membrane. So this has been called a soliton. It's a solitary wave, the localized wave, which is actually electromechanical solitary or soliton. That's interesting in its own right. And maybe at the end we'll be able to come back to it, because there's something quite intriguing and to my knowledge not really deeply explored, namely the membrane has phospholipid membrane has dipole moments in the head groups, and as you pulse send a pulse of electrical field, voltage gradient is an electrical field, these dipoles can respond to it and align to the field, at least temporarily, and then there could be a secondary signal propagation with a potential actually for electromagnetic wave emission from the axonal membranes. There are some papers which actually support this hypothesis that there are electromagnetic fields in between neurons and connecting across the brain, which would be quite interesting to see the secondary communication system, not just electrical, going in straight lines and across the synapse and activating and deactivating neurons, but also something which goes across the entire space inside the brain. Why is that important? Because that immediately allows to address some of the pressing issues, because electromagnetic waves propagate with the speed of light, which is 3 times 10 to the 8th meters per second, as opposed to electric signals would propagate at best, let's say at best would be, for the ease of calculation, 30 meters per second, which tells you that we have a 10 million time speed up of communication. And that's enormous. There are some open questions about how fast we can react to stimuli. I mentioned to you Libet's 500 milliseconds, but there are examples where this is truly hard to believe that it's explaining amazing speeds of reaction for top athletes, baseball pitchers and batters and hockey players, of course, and tennis players and all that. And also how fast we can, some of us at least, can perform some of complicated operations, such as solving, arithmetic questions with numbers of many digits and taking square roots of these and so on. Anyway, I don't want to dwell on this too much because this is kind of a phenomenology and some of it is quite speculative, but this is showing you how neurons are interacting with other neurons because they have a dendritic tree. So it's through dendrites that you form synaptic connections with other neurons. and we'll come back to it a bit later on. Neurotransmitters are being released from the presynaptic connection and travel to the postsynaptic, and then they trigger electric activity in the second neuron. What you see here also, I want to right away tell you, is protein filaments of great importance. Here we have actin filaments, and here we have microtubules, and we'll talk about this at length today because that is the basis of some of the most interesting theories about things like consciousness and quantum effects. So we actually now try in this talk to reconcile two enigmas, the mind being an enigma and quantum physics being another enigma, and maybe the two enigmas somehow are co-joined. And whether this explains any of the enigmas, maybe not, but it's quite likely that there is something in common here. And these are the most prominent scientists who explore these ideas, including Alan Turing, of the decoding of Enigma, the German Second World War German codes fame, as well as the information science godfather, let's say, Eugene Wigner and Karl Popper, Wolfgang Pauli that I mentioned before in the previous lectures, and Freeman Dyson, one of the greatest thinkers of the previous century, and John Nichols, who got the Nobel Prize for Physiology in 1964 with Huxley and Hodgkin. John Wheeler famously stated quip it from bit, or which means whether matter or information was first information. So it, matter, bit, information, or vice versa, bit from it. And then we're coming to our great physicists, Sir Roger Penrose, who got a Nobel Prize in 2020 for his work in gravity, cosmology and gravity, and he spent a lot of time, decades in fact, thinking about these big questions. And in fact, he unleashed a debate that is still going on with this book, The Emperor's New Mind, where he argued on a few hundred pages. It's a very dense book with lots of interesting comments, examples, intriguing statements about the the fact that the brain must inevitably be operating non-algorithmically, whether quantum mechanically or something else. It's still open. He argued for quantum, but definitely not classical deterministic operation of the brain, at least as far as the consciousness is concerned, is, according to Penrose, out of the question. This was written before the Nobel Prize in 1995 or something like this. I don't remember exactly. You can look it up. And that unleashed these debates I mentioned too, but actually preceding the book and continuing in parallel with the debates, Stuart Hameroff, who still collaborates with Roger Penrose, has organized these annual conferences on consciousness, consciousness from the scientific point of view. So they're called, they started being called Toward the Science of Consciousness, and now you can see it's This Science of Consciousness. I'm not quite sure if that is deserved or not. Let's say Science of Consciousness. This was in 2023 in Sicily. Last year, 2024, was in Tucson, Arizona, and this year in June, I believe, in Barcelona. Next year, we'll be back in Tucson, so it alternates between Tucson, Arizona, where Stuart Hameroff is a professor of anesthesiology, emeritus, and here, and a European destination, or sometimes, I think there were some conferences outside of Europe as well. All right, so what we are dealing with, and in the spirit of the previous lectures, I want to sort of zoom in and zoom out and look at, because quantum effects, of course, are mainly the domain of small things, but we argue that they can expand its presence and its role to bigger structures. So when you look at the neuron here with these dendrites, the soma, the axon here, you look inside, and what you see here is mitochondria. Of course, you need to power it with energy. So mitochondria provide, I would say, most of the energy, maybe not for the neuron. Neuron also uses a lot of energy from glycolysis. Mitochondria use oxidative phosphorylation, which, as we argued actually on Tuesday in the last lecture, is basically a quantum process. So there you go. You have already a process of quantum effects in neurons. And then you have bundles of microtubules inside the axon and also in dendrites. Importantly, they are oriented differently in dendrites and axons. And axons are unidirectionally. Here you have a bundle of microtubule. No, this is one microtubule. This is as you zoom in to microtubule, and microtubules have protofilaments, 13 protofilaments of tubulin dimers. 3 are shown here out of 13. And when you zoom in further, you see the building block of microtubules, which is a tubulin dimer with all the alpha helices and beta sheets. And so that's a protein about 8 nanometers in size, in length, and 4 and 5 nanometers in the perpendicular directions, width and height. This is credited to my former student, Travis Craddock, who is now a professor at the University of Waterloo. A great scientist, making important contributions to the field. And then there's some reactive oxygen species being produced by mitochondria and speculated to to give energy inputs into these possibly quantum states in microtubules, tubulin, and so on. But we're not there yet. Let's just explore some other things first. And microtubules, by the way, are interacting with ion channels, and they have been demonstrated to modulate or control even voltage-dependent ion channels. So they perform many roles, including being sort of gatekeepers to the cell. But what's important in this connection is the role in processes such as learning, memory, and learning. This is from a paper about 20 years ago, showing that microtubules are built and rebuilt continuously. And as you learn, let's say, a foreign language or a skill, or learn to play the guitar or something else, your cytoskeleton in the neurons undergoes reconstruction and rebuilding. And in addition to, of course, something called long-term potentiation, which is making synaptic connections, strengthening the synaptic connections between the neurons, which participate in some of these signals that come from the processes you work on, learning and memory and so on. And so that shows you a complicated architecture. It's constantly being built and rebuilt. And yeah, and here you see the microtubule cytoskeleton I tried to show before, which is really filling neurons densely. That goes back to Stuart Hameroff's work. As you can see here, Toward the Science of Consciousness. There was, at that time, 1996, it was called Toward, and there was a Science of Consciousness. Now it's the Science of Consciousness 30 years later or so. Let's leave it aside, just a side comment. And you see the microtubules here just drawn as little bold line segments, and they are connected by thin lines, which are microtubule associated proteins. And you can see this whole network. I want to give Stuart Hameroff credit for many observations he made over the years, which I think are fundamental. Namely that this resembles a computer architecture. And I think in 1987, if I remember correctly, he published a book. So that's now almost 40 years ago. I took up this book when I visited him for the first time. I met him in 1990 in Tucson, Arizona. And he just published this book a few years prior called Ultimate Computing. And it's still a fascinating read. At that time, I arrived late at night. He gave me the book to read. I couldn't sleep that night, to be quite honest. Ultimate Computing. And this was really an elaboration on his vision, which basically explains the potential for amazing computational power of the brain through the use of microtubules. And what's at that time wasn't quite clear how that computing could take place, but he made a lot of conjectures and hypotheses. And we have, time permitting, I'll get to some of that later today, and we'll get closer and closer to some of these predictions. So now you can see a little bit more microtubules connected by maps. And so the first thing I mentioned about this the changes here is that learning and memory strengthens synaptic connections, but also strengthens microtubule architecture inside neurons. So it's between neurons and inside neurons. And these are C-termini, very important protrusions, peptide chains sticking out of microtubules, which are loaded with electric charges, and they can possibly be sensing electromagnetic fields, and that could be one of the potential information processing centers, many of them, millions. Each microtubule may have about, depends on the length, but it will have 15 to 20,000 dimers of tubulin. Each dimer will have two of these protrusions antennae, let's say, if this is from the point of view electrostatics, electricity, an antenna, each one of these C termini is because of the charges there and responds to electromagnetic fields. So you'll have 30,000 antennae lining this one, and then several thousand microtubules. So you're talking about a system from the electromagnetic point of view, which has several million detectors of electromagnetic fields. Now you can imagine what kind of processing power this could have. Okay, so the challenge that has been taken up by my group and other groups was to try and see how this can be integrated across scales from the scale of a single dimer. We did a lot of computational work, experimental work on the conductive properties, electromagnetic properties. We're still doing it, interaction with electromagnetic fields, and published a lot of papers. I have no time to even list the papers here, which show some amazing properties of microtubules and tubulin vis-a-vis electromagnetic responses, and magnetic as well. And that's scaling up to a microtubule, and then you have microtubule bundles, and then you have interconnected axonal microtubules with maps. And also here you see the pathways to the synapse through actin filaments. So that architecture is incredibly sophisticated, I'd say, and can process information through various means. This is a real micrograph. Of course, the artist rendition here doesn't reflect the complexity, but it's kind of a microtubule tree inside axons. Okay, now, so that was on the positive side. The connection to neuroscience and to neurophysiology is that the cytoskeleton, especially microtubules, when get destabilized structurally or functionally, including trafficking microtubules through motor proteins, that is always linked to neurodegenerative diseases and traumatic brain injury, TBI. So there is a connection through tau, detachment of MAP tau through hyperphosphorylation to Alzheimer's disease, the deterioration of the motor protein traffic to Parkinson's. Of course, Parkinson's also is related to alpha-synuclein, and also there are connections to other neurodegenerative diseases such as Huntington's and Lou Gehrig's, stroke, and TBI. In TBI, there were some very interesting micrographs shown of broken microtubules in the brain of in the brains of deceased football players, for example, in the NFL. There was a very poignant movie about this. I forget the name, but the life expectancy of NFL players in the US is something like 50, was at least 53 or 55 years, something on this order. And then they discovered that many of them suffered from neurodegenerative diseases and succumbed to them. So, yeah, the cellular consequences of these repeated injuries to the brain were identified as broken microtubules and neurons, and that had upstream effects, downstream effects in terms of cognitive abilities and so on. So now the question is quantum mechanics relevant to the brain? Through these conferences and Roger Penrose's hammer of work, the answer seemed to be yes, but yeah, that requires big claims require big proofs. So we are not there yet. So is the brain a quantum computer? That will be incredibly powerful on top of already what I told you about the limit, the very small amount of power required and an amazing number of operations can perform in terms of classical operation, let alone quantum. That would be tremendous. And the next question is collapse of the wave function. You know, if it's quantum, then you could possibly draw a lot of consequences from it, including the collapse of the wave function at the level of, let's say, the whole brain or parts of the brain could be identified to something like a realized thought. You know, you have a lot of thoughts in your mind before you actually say something or do something. And before you do that, it's like a superposition of the wave function that we talked about in previous lectures. And once you decide, you know, you want to go for a bike or you want to eat a pizza or something, and then that's the thought that becomes now materialized, and that's like the collapse of the wave function. And then of course there's the flip side to it, namely if it's quantum, then we have the Heisenberg uncertainty, and that would give some limits on the predictability of the brain's functions, because you know the Heisenberg tells us that you can't predict anything, even a single electron's state. You can find out where it is, but you don't know how fast it's moving, or you know its energy, but you don't know when it had this energy. So how can you predict anything about the brain? Not anything, perhaps is too big a word, but precisely the brain's behavior. This leads us to some very big philosophical and ethical questions. If the brain actually is operating at the quantum level and it's not deterministic, is this where free will comes from? And if it's not quantum, it's deterministic, then we have no free will. That I leave this as an open question. Some people, like the Stanford professor of biology, Sapolsky, claim that we have no free will. I completely disagree with this. We do have free will. Whether it's unlimited is another question. I would say it's limited, but it's free to some degree, as everything else in this world. So that will have implications for artificial intelligence. If our brain is operating at the quantum level, then AI, if it wants to exert supremacy over us, it probably needs to go into this quantum direction to approximate the human brain's capabilities. And that's another topic maybe for another day, but everybody's talking about AI. I just want to say that I'm not as, well, it's fantastic. Yes, it's a great achievement. I use it every day. But it's more based on what the humans already created, and it uses to give us answers about things that have been generated. Knowledge, the body of knowledge has been generated before, and can mimic some things, but I don't think it has the yet at all even approximately the creative capabilities of the human mind. All right. So we have the functional unit of the brain, which is which is the neuron, and this is from Stuart Hameroff's early work in the 1990s. He looked at the microtubule and speculated the microtubules can have encoded states. So black and white will be classical states, and at that time, cellular automata were very popular as a new concept in computational sciences, computer sciences, and so he imagined that microtubules can perform some computation through behavior along the lines of cellular automata. That was before the quantum element was thrown in by Roger Penrose as a result of this collaboration, and that is a few years later, following the collaboration with Penrose, instead of binary cellular automata, so binary that's white square would be 0 and black would be 1. Now you have a qubit. Each dimer is in a quantum superposition state, so it's a qubit, and it has a wave function instead of just a digit, and that would endow microtubules, and therefore the brain with an incredible capability for quantum computing. Still to be demonstrated, and we'll come to it. And this has since become known as the Hammer of Penrose ORCOR model. ORCOR stands for Orchestrated Objective Reduction. Objective because they claim it's this collapse of the quantum wave function of neuronal microtubules. Here is the qubit, the protein qubit, tubulin qubit. The collapse of the wave function is spontaneous. It's not driven by anything outside. It's actually internally generated by interaction between the two states of tubulin. I will not go too much into this because that requires a lot of additional discussions about quantum gravity and so on. But let's leave it at this. So objectives meaning spontaneous, but orchestrated meaning that you have to have a sufficient number of participating tubulins in a given collapse of the wave function. So it's kind of a collective state, and that would make sense because, these thoughts that we talk about as representations or manifestations of collapse are across the domains of the brain. You may think about what I'm talking about right now. At the same time, you may be also thinking about what you'll have for dinner tonight or what movie you will be watching. So it doesn't have to be the entire brain, but some brain domains and objective reduction, orchestrated objective reduction, because the state is reduced to a classical state. I did some simple calculations about if this is all making sense, assuming hypothetically that these tubulin dimers actually do some information processing, then I did some calculations about how many states could be processed and how fast. What would be the memory capacity of the brain with these kinds of microprocessors? So microtubule is a microprocessor, microtubule. So this C termini I told you about could have 4 states per dimer. There could be some electron hopping transitions we argued in various papers. It is known that each tubulin dimer has at least 2 conformational states. They are bent. One is bent, one is straight. And also it can have a GTP or GDP bound to alpha and beta. So there's these states and some other ones, but let's say if we assume that there's 32 possible states per dimer, that leads to the calculation of about 100 kilobytes per microtubule or 1 gigabyte per neuron. And since we have 100 billion neurons, you could have 10 to the 20th bits per brain. That's certainly enough to encode the entire memory of your lifetime, because you live about, several billion on average. I think a human lifespan of, let's say, 84 years or something like this corresponds to, I'm just guessing the number, something like 5 billion, let's say 10 billion, so 10 to the 10th seconds. So if you were to encode 10 billion bits of information per second, then you could encode all of this in your microtubules of the brain. Yeah. So anyway, these are just very large numbers. Now the question is how fast you can have transitions taking place, and yeah, if nanosecond, that's really fast. Some of these transitions definitely are on this scale. Some may be slower, maybe microseconds, but at the fastest rate of nanosecond transitions, 10 to the ninth per second, then you could have something like 10 to the 29th or 28th operations per second, and implying the entire capability of the whole brain, all the states. So that's astronomically fast. You remember we talked about the IBM's blue gene with 10 to this 15th. Now this is trillions of times more. Hypothetically, this is what I would say, stretching it to a maximum. But still within the realm of possibility. So yeah, now of course, okay, this is a calculation elaborated here, let's say a bit less, more conservative, 10 to the 27 is still incredible. Now there are some additional limitations on this due to the power consumption. These transitions consume power. There's something called the Landauer theorem in information physics, which tells you that each bit of information costs one on the order of 1 kT Landauer theorem. So one bit of information to erase or create, one bit costs, has an energy cost, which is at least K is the Boltzmann constant, T is the absolute temperature in Kelvin, times the logarithm of 2. For those of you who are trained to think in kilocalories per mole, chemists and biochemists and biologists, this is about something like 0.6 kilocalories per mole. So if you multiply the rates of these transitions by this, then you get the energetic demand on the brain. Now, so this is out of discussion. It's would be way too much, but even with this, you get something quite impressive. Maybe that number. I don't want to dwell on specific numbers. Yeah, this is actually the maximum, and the maximum number of operations, bits per second, given all these numbers and the consumption of the human brain of energy, nutritional energy, glucose, and conversion into ATP. So however big or small this number is, it's way bigger than the currently constructed technological devices, computational devices possess. What is memory? Well, different definitions, but it's basically the ability to encode, store, and recall information. So these three things: encode, store, and recall. And the current paradigm is based on long-term potentiation, so synaptic connections. That's kind of like a functional definition, but how do you store? How do you retrieve? There's really no explanation for this. And different people have different ideas. Some theories about memory going back to pre-Bram, and Lashley talking about holographic memory in the brain, creation of holograms. Rupert Sheldrake, a controversial biologist, claims the memories are not even stored in the brain, but they're in some sort of information space. And then Mike Levin from Tufts University conducted some amazing experiments with worms, planaria, and demonstrated that even by decapitating these worms and then they regrow their heads with brains, they maintain the memory, which seems to indicate that memory can be delocalized throughout the body. I mentioned in the beginning of this talk. And then there is how much of the memory can we have. So I told you that by using microtubules as memory storage devices, we could have about 10 to the 20th bits. This actually is consistent with John von Neumann's estimate going back 75 years. Some other people made very modest allowance for memory, including this is a different Landauer, not the one of the Landauer theorem. That's in this paper, only 1 gigabit of information according to Landauer, which would be like 2 bits per second, which is ridiculous. That's now these days you can have terabytes of memory in a memory stick, and the human brain is so much bigger and has so many more connections and so on. So I would say that's a pretty authoritative person in the information science field, John von Neumann, one of the greatest scientists of 20th century. So yeah, I mentioned to you before that cytoskeleton is involved in memory formation, and we published a paper with Stuart Hameroff and Travis Craddock in 2012, which became actually quite popular and was cited and commented on by the Globe and Mail and Corriere della Sera in Italy and other countries, the Times of India and so on, where we explained the experiments on mice, which indicated that if you inhibit an enzyme called calmodulin kinase, which it gets activated by calcium, so it's calcium calmodulin kinase complex. This complex phosphorylates microtubules in neurons, and yeah, this is the paper, and we explained that this could be a method of encoding memory in microtubules of neurons by getting information from synaptic activation, and this was illustrated, I would say, beautifully by Travis, showing this is that enzyme. It's a hollow enzyme, hexagonal, and this is a different view of this enzyme. That's from above here, and this is sideways here. And you can see it's a motor protein too at the same time. It walks on the surface of microtubules, and as it does so, it phosphorylates its serine residues in the C-termini. This is an electrostatic representation of the enzyme with blue denoting positive charges and red negative. And we showed that it actually fits beautifully into the microtubule lattice. So there's a geometrical match. This is a microtubule lattice, a patch of it. And it shows how this enzyme, looking a bit like a snowflake, binds to a patch of a microtubule. And this is the complex it makes. And as it does so, it can phosphorylate up to 6 bits of information or 6 residues, and we called it the memory code, microtubule memory code, and calculated the number of bits of information that can be created this way and obtain again 1 gigabyte per neuron, which would then lead to 10 to the 20th maximum 10 to the 20th bits per brain with 100 billion neurons. And this shows you these patterns that could be emerging as well. This is a representation of this enzyme phosphorylating microtubule. That's a microtubule. The enzyme is walking on it, and and leaving a trace, which is that encode the memory. And then, so that's the article which we published and got quite a bit of excitement. So back to the question of quantum computation, I mentioned to you that really is a debate, I would say, started by Sir Roger Penrose, and then collaboration with Stuart Hameroff led to a lot of papers and a lot of debates. So one of the arguments that this could be quantum is that when you look at a tubulin in a microtubule, you find a network of tryptophans. These are tryptophan residues, of which there is 8 in the dimer. And this network can be photoexcited. So the electronic states become delocalized, and the quantum wave function can connect it all. And this is very similar to photosynthesis in this FMO systems in chlorophyll, very similar and geometrically and in terms of distances. And so we've done some studies on whether it's possible to make it a quantum system. And indeed, again, with Travis Craddock, who was my PhD student at the time, we simulated this and obtained quantum states in this network that lasted something like picoseconds. Not long enough, maybe, but they could be probably sustained a bit longer by energy, a continuous energy supply. So these are some results and some papers published, including Journal of Royal Society, and we called it the feasibility of coherent energy transfer microtubules, showing that this can occur, that there was very rigorous quantum calculation. In 2014, it took us about 10 years to do experiments and demonstrate that indeed happens. Science moves very slowly. A decade later, that's again from this work of Travis, showing networks of tryptophans which can be quantum excited by light and electromagnetic waves in general. And then in other papers, Travis and his collaborators, now 2017, showed how these excitations can propagate along microtubules as signals, quantum states, and that can be possibly amplified and upregulated to microtubule networks, and then eventually some pyramidal neurons and between neurons into brain domains and the whole brain. So there is, I would say, a sketchy scheme of things, or framework where you could see how quantum excitations can spread out by entanglement, basically. Now, what Sir Roger Penrose contributed to this goes in a different direction. He actually pointed to the gravitational effects on these quantum states and their entanglement, and that's a very bold hypothesis. I have to tell you that, and I don't know if I'll have enough time to show the slide, maybe I do, that so far there's no experimental evidence for this, for the quantum states being gravitationally affected and caused to collapse. But we talked about anesthesia, so I promised that we'll talk about anesthetics, and because anesthesia is, as I mentioned last time, is a very powerful mechanism that allows us to turn off and on consciousness. So if consciousness is a quantum manifestation of the human brain's activity, then anesthesia is manifestly a way of quenching that quantum state. And there's some empirical evidence that anesthetics do something to microtubules and therefore interact with tubulin. They reduce polymerized microtubules, and all anesthetics interact with tubulin. So out of 500 detected proteins that interact with anesthetics, only tubulin is affected, among 2% affected by halothane, one of the anesthetics, and 1% affected by isoflurane. So it's a very uncommon situation where anesthetics interact with proteins, and this one interacts pretty much with all of them. And again, Travis did for his PhD work on the binding of anesthetics to tubulin, found that anesthetic molecules interact with tubulin and bind to places very close to tryptophans. And remember, tryptophans form this network which can form a quantum state. So yeah, now we can see connection possibly, and that was actually again our collaborative work with Stuart and Travis, and we published this work in 2015 and showed that actually this interaction causes a shift in the frequency or the energy of a quantum state of these tryptophans, and the shift is proportional to the potency of the anesthetics. It actually, if there are any anesthesiologists or people trained, excuse me, trained in anesthesiology, they know about the Mayer-Overton diagram, which is a plot showing linear correlation between the potency of anesthetics and the partition coefficient. So how they interact with are soluble in oil, basically. So the more soluble in oil, the more potent they are. So, but we show here that there is something else going on, that they actually may, their potency may be correlated with the effects on quantum states of tubulin. And this is a further demonstration of this work by my former student, now Dr. Eric Zizi at the Politecnico di Torino, and showing you how anesthetic molecules dynamically they bind weakly, but they have a lot of binding sites on tubulin. In addition, they also affect membranes, as is well known, but also change the physical properties of the membrane. They decrease the bending modulus, so make it softer, and change the thickness and the distances between phospholipid groups. So there are physical effects, mechanical effects, and quantum effects. Now, back to the theory of Stuart Hameroff and Penrose, and I'll finish in a few minutes. But we need to talk about some experimental effects. So how do we detect quantum states? Well, first of all, this is just a comment on the fact that there are these papers, recent papers, showing that gravity doesn't affect quantum wave function collapse. A group from Italy, Frascati, led by Dr. Cucciano, and performed these experiments. We got some funding from the Templeton Foundation, and with Greg Scholes, who is a famous chemist that wrote some early papers on photosynthesis as a quantum phenomenon. And my former student, Arad Kalra, experiments were performed at Princeton, and with my colleague Aristide Dogariou at the University of Central Florida, we embarked on the demonstration of quantum effects in tubule and microtubules using two different experimental setups. One which used ultraviolet excitations, and one which used visible light, laser light. This was at the University of Central Florida with Aristide Dogariou. This is the wavelength of excitation, and then there was a detection, and we observed the extinction of re-emission of energy absorbed by the system of tubulin and microtubules after 2-minute illumination. That was repeated and resulted in delayed luminescence. So re-emission of light over a very long period of time, up to minutes, which is interesting because that would indicate that indeed, and that's laser light, so it's a single quantum state that could be generated in microtubules. And we found also that it was longer lived in microtubules than in free tubulin, unassembled microtubule protein. And that's also from this experiment. And then some additional experiments demonstrated that anesthetics delayed this, sorry, not delayed, but shortened the process. So the quantum states in microtubules were longer than in tubulin, but when you add anesthetic molecules, they become shorter, which would go in the direction of Hameroff-Penrose hypothesis minus the gravity aspect. And this is just a very quick rundown of the experiments at Princeton, where single photon counting was performed, and the constructs were a bit different. Microtubules were labeled with some molecules AMCA, which reabsorb photons, and because of this, that was published here, we were able to also determine the range of these quantum states, the correlations, how far they extended. So the long and the short of it is that we discovered that the lifetimes of quantum states excited by ultraviolet electromagnetic waves is about up to 5 nanoseconds. So you can see this is actually 1 million times longer than our computational prediction. So that's great, because we want it to be a long-lived state. And the correlation range was about up to 6 nanometers, which is roughly the size of a dimer. So the quantum state again was roughly all the tryptophan groups from the combined state. And then, yes, again, we showed that these two anesthetics, etomidate and isoflurane, decrease the lifetimes of quantum states. So so we have this, yeah, actually 7 nanometers. So we have gravitational collapse of the wave function, very unlikely. We don't see this happening. However, tryptophan excitations so far too short-lived, 6 nanoseconds, longer than photosynthesis, but not on the physiological scale, which would be a millisecond. We need to find a mechanism that increases the lifetime substantially. And the distances over which correlations are occurring are on the order of the size of a dimer, but not a microtubule. Maybe there's a way of connecting it across it. We don't know yet. So more experiments are needed. But we have some encouraging experimental confirmation of many predictions. So So yeah, maybe that I will finish with this statement that the brain seems to be a virtual reality generator. Maybe it has great potential for memory storage and perhaps quantum information processing that for sure is impressive. So I'll stop here and allow you to ask questions if there are any.
It's nice that there's new content. It's gradually moving and changing. Yeah.
Slowly, we started 10 years ago and there was no experiment at that time. Now we have experimental data which inch very slowly toward confirmation of some of these predictions.
Yeah.
We still have a gap to fill, but there is something to work with.
But I think maybe without that, you you probably remember this moment in our interaction early in the course when you gave a lecture on computational drug design, right? And I was upset because I wanted quantum biology. So if you didn't have any of your own studies in this area, I think it'd be hard to keep enthusiasm for it, maybe. It's so far ahead of others. Um, so I think it's important for your teaching that you have your own your own experiments, right? Yeah.
Yeah, yes. I couldn't agree more. And I think there's more enthusiasm than ever because I think this field has become a bit more closer to mainstream, a bit more respected by the scientists, neuroscientists, and others. In early days, it was kind of a fringe, where you talked about quantum effects in biology, and there was photosynthesis, and that's about it. Now, some very hard-nosed physicists are talking about quantum, including Christoph Koch. He's a very famous neuroscientist who admit that there is a room for quantum effects in consciousness.
Yeah. Well, I think this idea of quantum AI will also make things easier for you. There'll be some big high profile breakthrough there, I think. And then suddenly your work will be more celebrated than it was before. Yeah, that's my
Yeah,
point.
I hope this will happen while I'm still here.
Yeah. No, I think it will. I mean, I'm arguing, for whatever you want to define it, artificial general intelligence or artificial superintelligence, occurring within the next 3 to 5 years. So I think you'll be around. So, yeah.
Fingers crossed. But I have to, yes, you're right. Uh, in the spirit of singularity, this is accelerating. The progress is accelerating rapidly now.
Yes. Yeah. Yeah. That that's right. And so yeah, there'll be more new content every time. So that's good. Okay, well, thank you very much, and we'll see you next term.
Right. Take care. Thank you.
Yeah.
Thank you all.
Bye bye.